Good afternoon, everyone. We are coming back to our today's session, afternoon session. I am welcoming here our today's guest, Nannikanta Sahu sir, Dean and Faculty of Pharmaceutical Sciences, Rama University, Kanpur. Sir has completed PhD in Pharmaceutical Analysis. Sir has completed postdoctoral from Vietnam, FSASS, and SRAP are the special courses Sir has done, Statistical Reasoning and Algorithm in Pharmacovigilance, completed course in AI and Drug Discovery. Sir also has done drug review process, and Sir has many awards for his work. Best Research and Paper Award, Research Best Researcher for Zero Investment Innovation for Education Initiatives. Best Research Paper for Pharmacy ITAP Awards. Best Young HOD of the Year of the Award for Global Education and Corporate Leadership Awards. Recognized as a regular reviewer from Journal of Taba University of Sciences. And he is a lifetime member of following associations, IIFER, and lifetime member of Indian Society of Analytical Sciences, Asia Society of Researcher, advisor of National Forum of Pharma Institutions, lifetime member of APTI, lifetime member Healthcare and Biological Sciences Research Association, lifetime member Foundation Health Environmental Research, Institute of Scholars. All those sir has very good knowledge. So let us welcome sir to give his valuable insights on metabolomic approach for biomarkers development. Thank you, sir. So please,
Okay,
sir, you can
so
start now, sir.
yeah, my slide is visible to all of you. Is it visible to all? Madam, is it visible my
Yes, sir.
slides?
Yes, sir.
Okay,
Your slides are visible.
okay, okay. Thank you. Thanks for
Sir,
giving
can you
this.
make it full size, full screen?
Sir, make it as full screen, sir.
Okay,
Slideshow.
okay. Is it visible?
Okay, sir.
Is it
Yes, sir. Yes, sir.
okay? Okay. Thank you. Thank you very much for giving this opportunity. And I think I am audible and visible.
Yes, sir.
Okay. So thank you very much again, Malla Reddy College of Pharmacy, for giving this opportunity for this particular talk on metabolomics approach for biomarkers development. As you know, also before that, I like to thank all the organizing team, all the faculty members, all the registered participants for this particular session, and who are there really waiting for my talk. I was somewhat busy in some other meeting in the VC with our respected VC sir. So for that it was delayed. So sorry for the inconvenience to the organizing team as well as the listeners. But anyway, at last we are able to meet again in the virtual mode. And my topic today is on metabolomics approach for biomarkers development. So I'll not take that much time. I'll start immediately. Let me introduce about myself. Already she has introduced myself. I'm working as dean and principal in Rama University, and presently my work area is on my one of my research grant is approved on this metabolomics approach actually. So that is why I thought I need to share with all of you so that you may be provoked towards the research and innovations on this particular topic. So again, this metabolomics approach for biomarkers development is very important topic, as you know. Nowadays, with critical diseases, critical health issues are happening, we are unable to know the exact cause of that particular disease. It is happening suddenly. So my objective is to aware the people how we will be able to know the exact prognosis, exact what condition we are with the disease condition, so that we can be able to go for the treatment process. means we can go for the exact treatment process. That is what the objective of my study. So for that I have chosen this topic, and metabolomics approach is a very emerging field right now in the field of analytical chemistry. So first of all, we should know what is actually metabolomics, and this omics science is very very important. To nowadays, everywhere the processes are like your metabolomics, genomics, proteomics, and these types of techniques are right now happening everywhere and are implemented everywhere by the scientists and the researchers. Okay, so if we'll talk about this in my this particular means your talk, I have included like introduction, methods, applications, challenges and problems, future directions and conclusions. I have included like this. So first of all, I'll talk about your metabolomics. What is metabolomic science? Before going to metabolomic science, I like to study, I like to know, I like to tell you that what are metabolites. Metabolites, you know that whenever anything it is going to our body, externally to your body, it is undergoing the metabolism process. Okay, anything you are taking, breathing also, it is also inside your body. Metabolism process is going on. Without metabolism process, nothing is possible in your body. And during the metabolism process, many metabolites are formed, and those metabolites may be useful sometimes, and sometimes they are not useful also. So body is not accepting the not useful materials, not useful metabolites, and they are excreted out finally from your body through your either urine or through the excretory system. If it is not excreted properly, if it is accumulated in your body, then definitely it is going to harm your body. So this metabolomics is nothing but study of metabolites. है ना? We have to study the metabolites in specific your cells or organs in the body fluid wherever possible, right? So we are able to see the how many metabolites are there, and from that we are able to find out. We are by using certain techniques, we are supposed to find out whether these metabolites are going to be useful or they are going to be harmful for our body. And if they are not going to be useful, if they are harmful for our body, and if they are causing certainly some kind of disease, so we need to find out exact why, what kind of chemical or what kind of metabolite is particularly responsible for causing that particular disease. This is the main objective of this particular topic, that is metabolomic approach for the your identification of biomarker. Means we need to identify the biomarker, which biomarker is actually responsible for causing that disease. And if we'll be able to know the exact nature of the metabolite, then definitely we are going to develop such kind of drugs which can act against that particular metabolite, and it may diminish that particular disease in your root level. So that is the main objective of this particular study. Okay, so this is your omics. As you know, your body with molecular weight less than 100 1000 dalton, like your sugars, nucleosides, organic acids, and number of acids are there, lipids, steroids, all these are coming under that. You can see this particular omics science, it was metabolomics. It was coined in the late 1990s. If you check out the history of this metabolomics, it was coined, means it was discovered the name in around 1990s. And first paper based on the metabolomics was published in 1998 in the Journal of Trends in Biotechnology. Okay, one of the renowned journal. In that they have certainly published one paper. Okay, and the main the authors were, as you can see here, Oliver Wilson, and these authors were there. And in this metabolomics is nothing but study of metabolome is group of metabolites. Okay, and it was started decades ago. Means this particular metabolism, your metabolomic approach, it came into picture in the late means just decade before. Okay, and if we'll talk about this particular, at that time when we were able to find out this metabolomic approach, we used to take the help of technique, that is your NMR. NMR is more accurate, so NMR we used to take. Okay, how we are used to take the blood samples at different time intervals. We take the blood samples, and blood samples we used to analyze by using nuclear magnetic resonance spectroscopy after proper processing, because directly blood sample we cannot inject into any sophisticated instrument. So those samples have to be that is, those have to be your processed. Until they will be processed and they will be free of all these matrix materials, it cannot be injected into the sophisticated systems. Okay, so if you'll come see the omic world, you can see here different types of fields like your transcriptomics. All are emerging fields: transcriptomics, genomics, proteomics, metabolomics, epigenomics, pharmacogenomics. So many terms are there in this omic world. Okay, genomics, as you know, it is study of DNA. Transcriptomics, it is study of RNA in the RNA level. Proteomics, it is the study of proteins, different types of proteins and proteins labels. And coming to metabolites, you can see here sugars, nucleotides, amino acids, and lipids. If you are going to study these kind of compounds, then they are coming under your metabolomics, means study of metabolome. Metabolome is nothing but collection of your metabolites. Okay. Now, why metabolomics? Okay, it's a very important question. Certainly it is coming to our mind. Why metabolomics? You know, as I said, you anything, whatever it is happening right now, you are sitting here, you are breathing, right? You are breathing, you are taking oxygen in. So whenever you are taking oxygen in, also inside your body, your metabolism is going on. You know that your body is constituted of different organs, cells, tissues, all this, right? So those are, until unless they means there will not be any metabolism, then every organ will die, and due to multi organ failure you may die also. Okay, finally. So but this is the thing in our body. Suppose we are eating, so it is undergoing the metabolism process. Until that means you are taking the food, carbohydrate, protein, your any materials, your food material you are taking, it is undergoing the breakdown process, digestion process. So digestion won't be happened by the taking of your metabolism process, by involvement of different enzymes and all this. Okay, so anything, any physiological function going on inside your body, it is involving the metabolism process. Okay, so that is why we need to know, if metabolism process will not be proper, if, there kind of any kind of disturbances will be there in the body, then definitely your body will suffer to many diseases. So that is why we need to know the metabolomics process, what metabolites are going to be formed, whether the metabolites which are formed, whether they are up to the level, up to the mark or not, right? So now, in other words, you will see, not all changes or abnormalities detected in genome or transcriptome may be causing abnormality or disease. Silent mutations inside our body, silently in the gene level, sometimes the mutations are happening, we are unable to know. But finally, when it comes to the critical conditions, we are able to know, yes, something was happening in our body. At that time, it is not in our hand. We cannot take any kind of step to reverse that. Means we cannot treat ourselves. So in that case, it is becoming critical cases. Okay, like as you know, in case of cancer, we are unable to know the formation of cancerous cells. When it is becoming to the critical level, third or fourth stage, when we are able to know, that time it is not in our hand, or we are, and that moment we are unable to do anything, and ultimately we are losing our lives. So that is why this metabolomic approach is very very important step to save our lives and save millions of lives. Okay, now some more comparisons you will see. In your genomics, 40,000 genes can be assessed at a time, means we are able to analyze 40,000 genes at a time. And transcriptomics, if you see, 15 means 1 lakh 50,000 transcripts, means in the RNA level we are able to find out. Proteomics, if you see, around your 1 lakh your proteins, sorry, 10 lakhs of proteins at a time we are able to analyze. Okay, metabolomics, similarly 2,500 metabolites we are able to analyze nowadays. But previously, decade before, when we used to use NMR, That time we are able to analyze only 250 metabolites, but right now we are able to analyze at a time 2500 metabolites. Do you imagine how many metabolites are going to be analyzed by sophisticated instruments like your LCMS and all this? Okay, so that is the advancement we have done in the medical field. Okay, now this is the beauty of this metabolomic study. Means at a time we are able to analyze around 2500 metabolites. So how sensitive the instrument may be. So as you can see here, so many techniques we are using for this omic science, like your two-dimensional gel electrophoresis, matrix assisted laser desorptions or ionization techniques, time of flight mass spectrophotometric methods, nuclear magnetic resonance vector, gas chromatographic methods. So these are the different types of methods we are using. Okay, coming to metabolic profiling, how we are going to profile means quantify any metabolite. You can see here quantitative study of a group of metabolites known within or associated with a particular metabolic pathway. So that is called quantitative study. How many metabolites are going to be involved? So these are mainly requirement for metabolic profiling. If you'll come to one example, I'll just tell you some kind of drugs are there, like your non-steroidal anti-inflammatory drugs are there. If you take, we generally take these kind of drugs for in case of your arthritis problems. Okay, joint problems, any kind of these kind of problem, we are taking some anti-inflammatory drugs. Okay, so if we certainly use these anti-inflammatory drugs for longer time, then what will happen? It will affect our liver, and when it will affect our liver, definitely liver damage will be there. Your hepatotoxicity will be there, and hepatotoxicity will lead to the liver injury, and liver injury will may further it may be converted into the liver carcinoma. So that is why we need to check whenever we are taking any kind of these particular hepatotoxicity drugs, the patient's your biological fluid or anything should be under investigation, and we need to find out, we need to investigate whether they are certainly in safe conditions or not, or whether their liver is safe or not. That is what required here. Okay, so hepatotoxic drug, I have given some example here, like your hepatocellular, cholestatic and mixed. Okay, like some of the example I have given here: acetaminophen, allopurinol, amiodarone, na, and anabolic steroids, chloropromazine, clopidogrel, amitriptyline, enalapril. So all these kind of drugs are mainly causing your liver injury. So how it is happening actually? Whenever any kind of, suppose I am giving you an example, your acetaminophen. Okay, this is a hepatotoxic drug. Whenever you will take this as acetaminophen, in presence of these kind of enzymes in your body, it may be converted into an acetyl parabenzoquinone imine, NAPQI. So that particular product NAPQI, it may be either converted into acetaminophen glutathione or acetaminophen protein. So what they will do, they will bind to the proteins and they will increase the oxidant stress, and finally it will cause liver toxicity. So the main compound, as you can see here, NAPQI and acetyl parabenzoquinone imine. So it is mainly causing your hepatotoxicity. And how it is formed? As I said you, it is formed from your acetaminophen whenever you are taking the drug for the long time durations. Okay, so the what is the basic workflow for your metabolomic study? So first of all, if you see sample collection, treatment and processing. Sample collection means the biological sample. Whenever we are taking any kind of drug and we are thinking that it what kind of chemical it is forming or what kind of compounds metabolites it is forming within our body, to know the exact nature, what we have to do, we have to collect the sample, either the biological samples, then we have to process for the treatment, and after that followed by we are going for the different separation techniques. So what are the different separation techniques we are going? Like your gas chromatography, high performance liquid chromatography, ultra performance liquid chromatography, electrophoresis, and all this. Okay, Yes. Is it audible, madam? Is it audible?
Yes, sir, it is audible, sir.
Okay. Okay. And so, as I said, you in the basic workflow, in the basic workflow, you can say here, like sample collection, as I told you. Then we have to move forward for your treatment processing. Means we cannot inject the biological sample into the what you can say, into the directly into the instruments like your GC, HPLC, UPLC, all things. Okay, so we need to go for the processing. Means we need to take the we have to take the metabolites out of other matrix, means any extracellular materials, and after that it should be followed for the validation of the validation followed by clinical applications. Means after detection using this separation techniques, we need to go for the validation of those particular metabolites whatever they are, and after that we need to we have to go for the detection here by using the NMR or mass spectroscopic method, either NMR or either mass spectroscopy method. After separating out all the different metabolites, we need to go for the proper detection. Then we have to apply different types. So you know that 2,500 metabolites, suppose it will be detected, and 2,500 peaks will be generally identified in your that particular instrument. It is difficult to separate it out and to identify all these. So what we have to do, we have to apply different statistical tools. We have to apply different types of, you can say, the multivariate analysis, you can say MBA method, principal component analysis, PCA, partial least square methods, orthogonal PLS methods. So these methods, why these are applied to find to separate different types of signals into a same group. Means we have to separate all the signals into a separate. I mean, we have to separate it, and we have to finally identify. We have to validate all those biomarkers. So that is what the main basic workflow of any metabolomic approach. Okay. Now, as you can see here, I have told you the metabolomic samples, how you will collect these metabolomic samples. So, what are the different metabolomic samples you need to collect during the metabolomic process? One is your either you need to go for assessment can be pursued both in vitro and in vivo using cells, fluids, and tissues. So, you need to go for those. And biofluids are generally very easiest to work with. Generally blood samples, whenever you are having any health issues, so you are going to the doctor is telling to give you to give the CBP, complete blood picture. So complete blood picture, what happened, you they will take your blood samples with biological fluid, that blood samples. Then finally in by that blood samples they will process the sample, and they will separate out all the extracellular materials. Then they will inject into the different sophisticated instruments they have. Okay, so just for example, I'm just giving you to know the sodium level and potassium level in your body. They are using flame photometer. So similarly, we are using different types of sophisticated instruments like your LCMS, JCMS, NMR to find out all these kind of levels in your different types of lipids level and all this. So to use that, we are using this kind of sophisticated instruments. Okay, so that is why it is very easy to take your serum or plasma sample or urine sample to see the creatinine and all these, acetic fluid, pleural fluid, saliva, bronchial versus, and prostatic secretions. So these are different types of metabolic samples we are choosing. Okay, and next is your sample collections handling. After collecting these samples, how to handle these? These biological samples, after collecting, what we have to do, we have to go for some your handling process, how to handle those special requirements for diet, physical activities, and other patient validations. Due to high susceptibility of metabolic pathways, exogenous environment, maintaining low temperature, consistent sample extraction is very, very essential. Okay, so for biofluids, you can see standard volume how much we have to take, zero point 0.1 to zero point 0.5 ml for the analysis. For this metabolomic approach, how much of the biofluid is required? That is 0.1 to 0.5 ml. Okay. For NMR, minimum sample preparation is required. For NMR study, if you take very less quantity of sample also, it is enough. Okay. This is the basic flow as I have told you. Sample treatment and processing. We are following different types of separation techniques. Then we are following the different detection techniques, and we are able to find out the different peaks you will be getting here. Like your UPLC will be getting like this. In your HPLC you will be getting this peaks. Okay. Now after getting this sample collection, as I said you are going for the separation. Then you are going for the detection technique using mass and NMR. Okay. And finally you are going for the different application of your principal component analysis or different multivariate analysis, different statistical method to separate different same signals. Okay. What are the different ionization, different detection techniques? As you can see here, mass spectroscopy we are using. Whenever we are using the mass spectroscopy, we are telling it LCMS based biomarker development, LCMS based metabolomic approach for biomarker development. And suppose we are using in place of LCMS NMR, then we are saying it as LCNMR based metabolomics for the identification of biomarkers. So like this, different types of your identification techniques are there. Okay. So this is the technique, as you know, in the gas chromatography, the gas cylinder is there, it is executing the pressure and the mobile phase, it is executing, then it is passing through the column. We are just injecting the sample, the blood sample after processing, we are injecting into the column, and it is passing through the column, it is separated out, and finally it is detected by suitable detector. Okay. Maybe mass detector, maybe any detector will be there. Okay. Now this is the way how we are identifying the different peaks from the metabolite, whatever we have collected from biological samples. And when we are injecting into the system, SPLC system, so it is going to be identified like this. You will get peak like this. Okay. So this is the separation technique of HPLC. How we are using the HPLC solvent reservoir. When we are pumped to produce high pressure, sample injection is happened through the HPLC column, it is detected, it is separated, and in the detector it is detected, and finally we are detecting the peak. Okay. And based on the different types of peaks, we are applying different types of multi-component analysis, and we are separating different types of materials based on their nature. Okay. This is the difference between your SPLC compared to UPLC. UPLC you may be saying showing it is number of peaks are going to be observed here. It is very sensitive. Okay, in HPLC some specific peaks are going to be identified. So UPLC you can say it is more sensitive than your HPLC. Okay, and in your detection technique NMRS doesn't depend on the separation, relatively intensity NMR spectra difficult for interpretations, applicable in MRI. Okay. So in your NMR spectroscopy, we are going to find out the exact nature of the nucleus of the particular metabolite. We are able to find out the nature of protein in that particular compound. Based on that, we are able to find out whether it is shielded or deshielded. The proteins are shielded or deshielded, and from that we are able to know the nature of the environment. Okay, whether it is a methylene proton, whether it is a methyl proton, or whether it is a ethyl proton, what kind of proton it is. Okay. So this is the detection using NMR as I have shown you. This is the detection using NMR. Okay. So now, in case of detection techniques in your mass spectroscopy, the sample when it is injected, different ion sources are there, mass analyzers will be there. Finally, it is detected, and based on the charge by mass ratio, the charge by mass ratio, the compounds are going to be detected by using your mass spectrophotometer. This is the difference between NMR and mass, and you can see NMR is more sensitive than mass, but one drawback with NMR is that it is able to identify very less number of metabolites, that is 250 maximum. But in case of your mass spectrophotometry, it is able to identify number of peaks, number of compounds. Okay. So these are the metabolites detected in cancer by NMR. In your cancer disease, different types of metabolites are there, and they are going to be detected. So till now, some kind of okay, so different types of metabolites detected in cancer by NMR till date, like your leucine, isoleucine, valine, glutamate. So certainly these kind of compounds sometimes they may cause your different types of cancer in our body. Okay, uh, in your prostate cancer some kind of are there, like you have seen in your liver carcinoma. Your napui is one of the metabolite which is responsible, right? So like that, different types of compounds which are detected till date as biomarker for this cancer by NMR. Okay, so how to profile metabolite profiling? So metabolite profiling, how we have to go for, as I said you, by using this kind of sophisticated techniques. Okay, general approach I have already told you how we are going from the biological samples we are taking, like your human and animal fluids and tissues. Even you can take the yeast or bacteria, cultured cells, growth material, media, and we are going to go for the analytical profiling, processing process steps. Then finally, we are applying the statistical methods, and finally, we are screening out all this process by using your this kind of principal component analysis and all this. Okay, so this is the way how we are observing. See, two types of metabolomics is of two types. One is your targeted metabolomics and another one is your on-targeted metabolomics. So targeted metabolomics is nothing but, you know that certainly these kind of metabolites, suppose xyz is causing the disease. So what you have to do, you need to you need to target xyz. You know that xyz is causing the disease. Suppose, as I said, you identified biomarkers, that is your leucine, suppose one of the biomarker. Okay, you know that leucine is causing the cancer. So you need to right now quantify the leucine level. Okay, so that is what I'm that is called targeted quantitative metabolomics. Okay, on targeted means you don't know which group of metabolomes are causing the disease. You don't know, you want to find out. So certainly what you have to do, you have to take the biological fluid, you have to separate it out by using different types of techniques like your HPLC or whatever. Then finally you have to go over the detection technique using mass spectrophotometry, and finally you have to detect the compounds. Okay, so this is what about your on targeted metabolomics means, when the metabolomics are not known to the analyst, means we don't know which metabolites are causing the particular disease. Okay, so this is the process. So what we have to do, we need to, whenever any metabolomic study we used to do, what we have to do, we have to take the sample from a standard, means anything we need to compare with a healthy patient, to person. So with a healthy person, the patient, from the patient also you have to take the sample, and from the control group, means those who are not having any disease, from that also you have to take the biological sample. This is from the disease group, this is from the control group, those who are not having any disease. So we are taking the sample, either tissue, plasma, urine, saliva, cells, all these we are taking. Finally we are going for the sample preparation, means we are separating the metabolites. How we are going to separate out, we are separating, or we are processing the biological sample based on different types of method. One is your liquid extraction, one is your solid phase extraction, and another one is your micro solid phase extraction. So different types of techniques are there, by virtue of which we are separating the samples, or we are separating the metabolites from the extracellular materials. Okay, So that is what called your sample preparation method. You know that other foreign compounds are also will be there in conjunction with your the metabolites. So we need to separate out the metabolites. Until and unless we will not be able to separate the metabolites, the peaks for different the impurities and all this will be found out, will be detected, and finally it will be very difficult to handle those to analyze the compound. Okay, so that is why sample preparation and processing is very very important. Okay, now sample can be analyzed by using, as I said you, GCMS, LCMS, capillary electrophoresis, mass spectrophotometry. So after that, it is from the group 1 and group 2, means from the disease group whatever the peaks we have got, from the control group whatever the peaks we have got, we have to compare by applying different types of data analysis methods, like your principal component analysis and all this. So it will separate different metabolites, different same nature metabolites into different groups. It will give different peaks there. Okay, suppose this group is for this kind of metabolites, this group is for this kind of metabolite, and this group is for this kind of metabolite. So you will get different types of peaks for all those. You will get, and from that easily you can identify, yes, this is the main group of metabolites which are causing the disease. Okay, so this is the very best way to know the exact condition of the disease, means we are able to know where the particular person is, the particular health condition is. Okay, this is a metabolomics workflow, already I have told you, right? Biological samples, data acquisitions, means we have to find out, take the biological samples, then data acquisition by using LCMS, GCMS, NMR. After getting all these spectra, we have to detect it by spectral processing methods. That is your noise filtering, peak detection, peak alignment, or normalization. So by virtue of this, we are going to analyze the data. We are applying some univariate analysis. You can see here, or multivariate analysis, and we are getting different types of metabolite identification. We are identifying different metabolites. Okay, and finally, what is that? Biological interpretations, means this particular metabolite is cross-linked with which kind of metabolic pathway. So, which metabolic pathway is able to produce these particular metabolites? For that, these metabolites are causing the disease. So, this is the main intention why we are using this metabolomics approach. And if we'll be able to know the pathway of production of those harmful metabolites, definitely if we'll stop the pathway formation of those harmful metabolites, the disease can be checked. The disease may not grow anymore. So that is what the main objective of this metabolomic approach. Okay, so these are the different peaks how you will get by using different types of detection technique. If you'll get, you are using 1D NMR, so you'll be getting a peak like this. If you are going to use this one, we are in a zoom conditions. So in the zoom condition, the same peak we are observing here, we are showing you here. And if you'll go for the LCMS, you will be getting a peak like this. Okay, in the mass spectrum, the peak will be like this. In the total ion chromatograph, the peak will be like this. Okay. Now, after getting this peak, we are supposed to we are supposed to use different assessment techniques. Okay. Goal is to reduce the time between collection and freezing the body fluids and tissues. Okay. Now, sample storage and shipment. Whenever you are getting the sample, the biological sample, you need to immediately store it into any cold, any refrigerated conditions, deep freezers. Like you need to maintain around minus 80 degrees Celsius. Okay. Then finally, the body fluids and cells can be kept as intact samples. If you are supposed to take any body fluids, or you are taking, we are going to take any body fluids in the sense like the saliva, mucus. है So these samples, whenever you are going to use, then you need to keep it in your intact samples. You as intact samples. Then good time to prepare specifically codes needed for analysis. Then you have to prepare the samples in the nano range, or in the micro level, or ppm level, or nano level also. Okay, this is the beauty of this metabolomics, that in nanogram level also you are able to nanogram, nano range also, nano concentration also, you are able to analyze the compounds. Okay, suppose you are going to take the cell extracts also, you are able to take the cell extract. Short term storage, store as extract in solution, and if it is for long term storage, then you need to evaporate the solvents and store only dried materials. Okay, so this is the way how we are going to handle the sample and shipment. Okay, now different types of methods are there. As I said you, solid phase extraction is there. It's solid through solid phase extraction. You need to pass through the specific column used for the preparation of specific metabolites. So this is the specific column. You're packed with different types of stationary phase. Finally, whenever we are allowing the sample to pass through, the samples will be separated out based on their retention time towards the stationary phase in the solid phase columns. Okay, cartridge, you can say cartridge, we are using the cartridge. So by what will happen, the unnecessary materials will be retained and the required materials will be retained, the unnecessary things will be filtered out. This is the way, and finally we have to collect this one and we have to process it and we have to inject into the your HPLC column to find out what exact nature of this particular metabolite is. Okay, this is your solid phase extraction. Liquid extraction, similarly, you need to separate the sample, the mixture of components to be analyzed by passing through, by taking one aqueous and one organic phase. That is called a liquid extraction. Between two liquids, you need to extract in which solvent it is going to be spreaded out. That solvent you need to find out. If in which solvent it is going to be more solubilized, then the solute content will go to that particular solvent. Then that solvent you need to filter it out, you need to separate it out, you have to evaporate and you have to find out the you have to go for the rotary evaporation and you have to find out your the required metabolites for the study. Okay, and coming to protein precipitation, as I said you, protein precipitation you need to go for either different types of salts we have to use. Add accurately measured volume of all organic solvent containing one or more internal standard and accurately measured amount of biological sample. Allow proteins to precipitate. So you need to go for the precipitation of the proteins. Okay, then after precipitation method, you need to go for centrifugation. And after centrifugations, you have to collect the supernatant liquid. And at the bottom, the protein which are precipitated right now, those you have to separate out and you have to make a proper solution, proper aliquot, and you have to inject into the sophisticated instrument. This is the preparation. This is the way how we are going for the protein precipitation methods. Okay. This is the tissue homogenization. If you are going to suppose to take any tissue to know whether they are means the tissue is what is that you can say whether it is infected, it is diseased. So you need to find out. In that case, whenever you are going to study the tissue, you need to take this tissue. Then you have to go for the pulverization. After that, you have to grind using mortar and pestle, and finally you have to go for the probe sonications, and after probe sonications you have to go for the focused acoustic waves, then bead beating. So you have to go for all these steps to separate the different the metabolites obtained from the tissues. Okay, you can see here I have already told you, in case of metabolomic approach you need to you have to take the biological samples from the diseased persons and from who are under treatment and the person who are not having any disease. So with that, you have to take the samples. You have to inject into LCMS. You are getting the peak like this. Suppose for the sample obtained from this condition one, you are getting peak like this. From the red one, you are getting the peak like this. Though these peaks are right now separated out, you have to process the data. Finally, you have to apply the statistical data. You have to go for the recursions. Finally, you have to go for the biological matrix separation out. And before that, you are going to find out, you are going to identify based on their mass by charge ratio. So this is the one, this is the way how we are going to proceed for this particular this metabolomic approach. Okay, so body fluid or homogeneous media LCMS sample, as you can see here, if it is a lipid samples, body fluid or homogeneous media 10 microliter is enough if it is lipid. And extraction solvent, what kind of extraction solvent you have to use? You have to use your isopropanol, that is 190 microliter. If we'll take 190 microliter of extraction fluid and body fluid sample, if we'll take 10 microliter, it is enough, right? How many sufficient quantity of metabolites you are going to identify. Okay. Next is your free fatty acids, bile acids, and all this. For that, 30 microliter is enough. And extraction solvent, you have to use your methanol. And if the compound is of nature, that is your nitrogenous compounds or cationic species, then you need to take it by using the solvent acetonitrile and methanol. And if it is going finally the sugars, organic acids, all these, then you need to select your methanol extraction, and you have to take 30 microliter. It will be extracted out. So this is the way how we are going to find out the different types of metabolites present in your body after a thorough metabolism process. Okay. The workflow already I have told you: sample preparation, checking, quantifications. Already I have told you the experimental designing, that is the sample preparation. Then you have to go for the NMR raw data, then spectral processing, data processing, multivariate analysis. Finally, you are applying the multivariate analysis. Finally, you are getting two such types of peaks, two types of results, like the supervised methods. Another one is your supervised method. One is your unsupervised method. Another one is your supervised method. Supervised method, we are using the technique like your PLSDA, OPLSDA. But in case of unsupervised, we are using your PCA, principal component analysis technique. Okay. So this is one of the research article published. So right now coming to the last part, that is your data analysis and interpretation. Already I have told you, first is your data collection and sampling method. After that your data processing. Data processing, after that what you are doing, your instrumentation method, analysis method, or separation method by using your HPLC, UPLC, all these NMR, we are going to separate it out. And finally we are going for the detection method using your suitable detectors. Okay, and finally after detecting, now the samples have been separated out. The next step is your, the samples have to be analyzed. The samples, how they will be analyzed? They will be analyzed by your different bioanalytical tools, bioanalytical software, biostatistical software. Like one of the proper tool is there, that is your principal component analysis. So by using principal component analysis, PLS method, OPLS method, we are able to analyze this data, whatever it is obtained after thorough process of your this one, your separation process. Okay. Now coming to data analysis in the step one, as you can see here, formation spectral data set for pattern recognition. In the data analysis part, you can see here, suppose I am telling you that is your NMR or mass spectra for biofluids, tumor tissue containing hundreds of signals from endogenous metabolites, converted to spectral data sets, reduced to 100 to 500 spectral segments. Okay, and their respective signals intensities are directly entered into the statistical program. So whatever the peak and graph will be there, you know that by using the specific detector, it is separating out the different groups. You can see here CIML non-treated, CIML resistant, one is your CIML imatinib. With CIML, we are treating with the drug imatinib. One is your CIML non-treated, means this is without drug, and another one is your CML resistant, means certain kind of drugs we have given, but it is not responding. So this is called, these three types of peaks it is separating out. This is what I said you, the separation and segregations of the different types of peaks. Okay, this is the data analysis. As you can see, step 2, linking specific spectral region. So after finding out the exact, you can see here CIML imatinib, CIML non-treated, CIML resistant. So these three types of substances are separated out right now. Finally, we have to analyze from there individually. We have to apply here, and we have to know which metabolite is going to cause the disease in your body. Okay. This is the step 3. You can see quantification and association of putative biomarkers with respect to particular characteristics or outcome. Okay. Now, so these are the different methods, as I told you: data analysis and interpretations. So principal component analysis. sound independent modeling of class analysis, SIMCA, one of the method is there. Partial least square method is there also. Orthogonal PLS method is there, OPLS. Another one is your target profiling. So these are the different techniques by virtue of which we are by virtue of which we are going to use, or we are going to analyze the different peaks obtained by using different your special techniques or specific techniques. Okay. So now in the proton, already I have told you, your principal component analysis is one of them. It is actually unsupervised. It is following with the technique that is a multivariate analysis based on the projection of the methods. Main tool used is your chemometrics. So we are using one of the tool that is your chemometric. Okay, you might have seen some of the method like your chemometric assisted method development of so and so. So like that, here one supporting chemometric methods are there by virtue of which that we are able to separate the different metabolites based on their nature, and we are means separation and segregation different are there. Okay, and finally we are going to identify the nature of the metabolites. Okay, now we are alerting different scores also to those metabolites. Okay, one of the method is there, your SIMCA techniques also there. SIMCA technique is nothing but your supervised learning method based on your PCA, principal component analysis. Okay, what we are doing here, construct a separate PCA model for each known class of observations. Each known class of observations, for that we need to go for the construction of the PCA graph, PCA model, principal component analysis, then boundaries defined by 95% class intervals. Okay, recommended for use in one class case or for classification of any interpretation methods. So these are the different types of SIMCA techniques are there. One class problem I'll just share with you. Only disease observation defined a class. Control samples are too heterogeneous. For example, due to other variations caused by disease, gender, age, diet, lifestyle, etc. So this is a becoming a barrier in that, and this SIMCA techniques is also there, but it is not that much advanced like your principal component analysis. Okay, one other method is there for the data analysis, that is your PLS method. Okay, PLS method. So in the PLS method, what we are doing, these are the supervised learning method, and here recommended for two class cases instead of using SIMCA. So here two class cases we are going to analyze by without using of any SIMCA. Okay, so principle that of PCA, that is your principal component analysis, but in PLS a second piece of information is used, namely the labeled set of class identities. So here we are using a second label, that is your called labeled set class identities. Then two data tables considered, namely X, input data from sample, and Y, containing qualitative values such as class belonging treatment samples on that. Okay. So these are the various method, as you can see here, by virtue of which we are going to separate the different signals. Okay. These are some of the list of the tools we are using for analyzing some of the metabolites, like MetaboAnalyst 2. This is very widely used metabolomic software. That is MetaboAnalyst 2, like MadeDart. You can see here MadeDart, then MetaboMiner, then Camera, MetaboHunter, then your Okay, so these are different types of tools we are using for identification of the purpose. Okay, these are the things. Targeting profile already I have told you. Databases, large amount of data, need for databases that can be easily searched, newly emerging databases. So all these kind of techniques are there. And after that, we are coming to that summary of metabolic database. If you speak, we are using certain tools also, software tools also, like your human metabolome database. One is there. In that database, we are able to find out different types of biomarkers and all this by using this HMTB. Okay, like Biomag, Risk Bank, and PMRB, that is called metabolomics. So this is also used in case of NMR based metabolomic study. Then BCG, that is your database of biochemical, genetic, and genomic metabolomic network. Okay, BIGG. What is that BIGG? BIGG is nothing but biochemical, genetic, genomic metabolic network. Okay. Now, Fine metabolome database, Metlene metabolite database, NIST spectral database. So these are various kinds of metabolomic databases available, and we are using those. Out of that, one of the important database is your HMDB, Human Metabolome Database. By using this Human Metabolome Database, you can see here what is happening. The text synonym speech, whatever the things are there, it is going directly to that. Structure search, you can use this particular HMDB tool for the structure. For your molecular weight, we are using this one particularly, the molecular weight search. Then substructure search also. By using this HMDB, this tool, the data, whatever you have data set you have obtained by using different types of techniques in the analysis and in the analysis technique, by using this tool, you can be able to identify this kind of particular metabolites. Okay, and their different natures, molecular formula, structure, molecular weight. Okay, they are mass spectroscopic spectral search. So everything you are able to find out by using this particular HMDB software, that is called human metabolome database, that is called HMDB. This is very important database. Okay, what are the different applications of this metabolomic disease? In case of drug discovery also we are using, in toxicology we are using, in case of your markers of disease, biomarker development we are using, newborn screening we are used to do by taking the embryo to know the nature of the embryo. We are going to identify food safety and transplant. So everywhere you can see the applications of your this particular technique, that is your metabolomics approach. And of course, pharmacology and preclinical drug trials, your toxicology, transplant monitoring, newborn screening, clinical chemistry, true for functional genomics. So, lots of applications are there in case of IVF or in vitro fertilization. So, in that case, also we are using this particular metabolomics approach technique. Unnecessary, those who are which are not of good quality embryo, we are just segregating out by using this, by applying this particular metabolomic approach. So many applications are there. You can see, you have seen also in the oncology field also we are able to use this biomarker development, because as I said you, if you'll be able to know the exact disease condition, then only you can be able to cure. If you'll not be able to know the exact disease condition, you cannot go for the proper treatment approach. Okay. So in the diagnosis, that is your carcinoma prostate making difference in case of this. This is one of the example I have given just only for your knowledge enhancement. So these are one of the your paper published by this related to prostate carcinoma using this metabolomic approach. Okay, one of the material is there, sarcosine, one of the material. That sarcosine can be detected using non-invasively in the urine. In the urine, we are able to identify this sarcosine. If we take any patient who is struggling with your prostate cancer, then in the urine samples we are able to find out this sarcosine's level. This sarcosine, based on the sarcosine level, we are able to find out the exact disease condition. So sarcosine is nothing but it's a disease biomarker. Okay, so there are so many examples for that. If possible, with the participants upon their request, I'll share this PPT. In that, I have included so many cases, case studies, and some of the examples. So long answer. Similarly, some compounds are there which are responsible for causing your lung carcinoma. Okay. Sometimes what is happening, where breath pattern of exhaled breath volatile organic compounds, wherever any pollution is very heavy, so there we used to use some of the filtration unit. If we'll not use that, then harmful pollutants may be inhaled by ourselves, and it may cause some. It will move towards your lung carcinoma. Okay, so you know that accuracy exceeding your 80% in lung cancers. Okay, so these are the different types of applications of your this metabolomics approach. Similarly, in case of breast cancer, also you are using this particular your technique. That is one of the example I have given here, NMR and NMR based your spectroscopy. Okay, so one of the choline detection. So choline is mainly responsible for this breast cancer. So we are able to find out this choline level in the breast, so that we are able to find out this breast cancer conditions and all. Okay, so in case of ovarian cancers also, due to happening, sometimes it is happening because of some kind of your PCOS, polycystic ovarian syndrome, leads to your continuous due to hormonal imbalance, and finally sometimes it is may causing your cervical cancer. In that case, we are able to find out all this. Okay, so some of the metabolomic your biomarker which are identified, some cancer biomarker, it is already I have seen shown here, like your alanine, it is causing your hepatoma and brain tumors. Okay, like your saturated lipids, it is causing your malignancy and all this. Okay, in case of glycine also, it is causing your decreased following disruptions of HF1 pathway. So like that, your myoinositol increased in colon your cancer. It is causing your colon carcinoma. Okay, myoinositol. It is one of the biomarker developed till date. Polyunsaturated fatty acids. It is causing increased in gloma cells undergoing apoptosis and all these. Taurine, one of the biomarker is there identified till now, which is also causing your prostate cancer. So these are different types of biomarkers you're responsible for different types of disease, and with this these are different applications I have included. This particular metabolomic approach, as I said you, this is a very novel method to identify the nature of the harmful metabolite, the harmful cancer biomarker, and we are able to know the exact condition of this particular disease, and we are able to take the step, means how we'll go for the treatment process. So with this, I like to thank everyone for your patientful listening, and I like to thank the organizing team for giving me a chance to deliver this talk in presence of the beautiful audience of not only of your college, of different audience, different platform, who have joined today and who are listening to me since 1 hour. So thank you very much to the principal of Malla Reddy College of Pharmacy, the convener of this particular faculty development program, and I like to thank also the participants, the management of our also university, our chancellor sir, our vice chancellor sir, our respected all the dignitaries, all the deans and heads of all our departments. Thank you very much. And if any queries are there, I'll be happy to answer all the queries from
Thank
the
you, sir, for your valuable insights. Thank you, sir, for valuable insights. Sir, one question is there, sir.
yeah, please.
Is any standard books are available for this topic?
Yes. For this, one of the bioanalysis book is there. It is a bioanalysis book. The exact author name I have forgot, but
Okay.
later I'll share with the author names of that particular book. In that, or you will say you'll tell me, so I'll share the e-books also to your email, so that you can share with all the participants.
Okay, sir. Sir, one more question. Sir, you can see the question in the comments, sir.
Okay, where it is?
Please open the comment.
Yeah, yeah, yeah. Right, right.
Can metabolomics be used?
To identify potential biomarker for disease that are currently asymptomatic or have non-specific symptoms. You see, that is why we are going to. It's a very good question. You have some of the participant, I think, has asked that is your means whether any disease is there. I think, sir, can you please mute somebody? I think somebody is talking. Uh, yeah. So fine. So one of the
Thank you,
question is
sir,
there.
for your
Yeah,
valuable insights.
yes, some diseases are there. Those which are asymptomatic. Asymptomatic means these are without any symptoms. So in that case, what is happening? Doctors are, we are doing right now every 6 months. That is why doctors are telling every 6 months we need to have a health checkup. So whenever we'll go for a health checkup, then only we'll be able to find out any kind of abnormalities in our body. So that is why we need to go for this one. This metabolomic study is very, very important. Okay. So see, timely diagnosis is very important to know the prognosis of the disease. If we'll not be able to diagnosis will not go for the diagnosis of the disease in time, definitely it will reach to certain level we cannot return back, and we'll face the casualty
Yes.
or anything, right? Yeah.
Sir, one
Okay.
more question is there.
Multiple metabolites can
One-
identify. Yes, we can be able to identify multiple metabolites. As I said you, around 2500 metabolites at a time we are able to identify by using this particular technique. Okay. But out of that we need to apply which are mainly causing disease. For that we need to apply the tools, the statistical tools, like your PCA, principal component analysis, multivariate analysis. From that we can segregate out the unnecessary metabolites, means those which are of no use. Okay. Sometimes, as I said you, the metabolites which are, some are useful for our body, so, and some are not useful for our body. So those which are not useful for our body, those need to be detected. That is what important.
One more
Yes,
question is
any
there,
other
sir.
questions? Yes. How? Okay, somebody is asking machine learning and artificial yes, definitely this is this metabolomic tools we are, whatever we are using, this PCA and all these, those are also included in your artificial intelligence. As I said you, one of the software, that is your tool, that is your HMDB, human metabolome database. Okay, so that is one of the important artificial intelligence tool, by virtue of which we are using, we are going to identify the nature of the metabolites. We are able to identify the biomarker. Identifying biomarker is not that much important. Identifying biomarker as well as validation of biomarker is very, very important. Okay, you need to validate the biomarker. Unless you will standardize the biomarker, you cannot come to the conclusion whether you, sometimes that body physiology is different. I mean, sometimes time to time it is changing. So maybe today the metabolite is formed, tomorrow may not be formed. So that is why you have to repeat it. You have to see this particular metabolite is formed or not. So you have to validate. That is called validation, repetition of the results. Okay. When you are going to get the repetition of the results, we are going to where you are going repeatedly the same kind of biomarker, then you can say yes, it is validated. Right now, this biomarker is responsible for that particular disease. That is what. Okay, machine learning. In fact, you can apply the machine learning, the coding part, the different programmings. This is coming under also artificial intelligence. That is your artificial neural network is there, right? So these are all part of artificial intelligence, and in fact, HMDB is an important artificial intelligence tool.
Okay, sir. No more questions. Thank you, sir. I thank behalf of Malla College of Pharmacy for
I
sharing
should be
your
thank-
insights on this
should
topic, sir.
I should be thankful to all of you, because I have made you wait for half an hour or 1 hour wait for me. I was busy with busy vice chancellor meeting hall. I was there. In fact, I could not give you time in a proper time. So I'm very much regret for that. I'm very much sorry for that also.
No, sir. With
Okay.
your topic is very valuable, sir. Everyone
Yeah,
is
yeah.
appreciating.
Yeah,
Thank
yeah,
you, sir.
thank you very much, sir. I was working whenever they are nearby your Malla Reddy College of Pharmacy in Mari Lakshman Reddy Institute of Pharmacy. I worked for around 6 years, and almost in South, means Hyderabad. I was for 13 to 14 years. So I love to interact with the people of that region, not only our Hyderabad, Andhra Pradesh, and all this. I like to thank. I like to interact with all the participants. Yes, thanks for your comments and for your valuable comments. And in future also, I'll be happy to share of my research work with all the people if we'll give a chance again. Thank you.
Thank you, sir. Okay, tomorrow on same session we are having a fourth day first session. So thank you for all.